When a cell is under stress, such as from DNA damage, a viral infection, or growth factor deprivation, it may undergo programmed cell death, also called apoptosis. In mammalian cells, many cell death signals trigger apoptosis through a pathway involving mitochondria. In the mitochondrial pathway of apoptosis, death signals within the cell activate the pro-apoptotic effector proteins BAX and BAC. When activated, BAX and BAC form oligomers in the outer membranes of mitochondria. Formation of the oligomers leads to the release of cytochrome C from the mitochondrial intermembrane space, either by forming pores or by interacting with other mitochondrial outer membrane proteins. While in the mitochondrion, cytochrome C participates in cellular respiration, but in the cytosol, cytochrome C drives the cell into apoptosis. Cytochrome C binds to the cytosolic proteins called APAF1. With cytochrome C attached, the APAF1 proteins can assemble into a structure called the apoptosome. Another protein, called caspase-9, is now activated by forming a complex with APAF1 in the apoptosome. Caspases are proteases that have cysteine residues at their active sites and cleave after aspartic acid residues in their substrate proteins. Two types of caspases play roles in apoptosis: initiator and effector caspases. Initiator caspases, such as caspase-9, are the first caspases activated by apoptotic signals. The function of initiator caspases is to activate effector caspases, such as procaspase-3, through proteolytic cleavage. Caspase-9 cleaves procaspase-3, producing active caspase-3. Caspases, like other enzymes, act catalytically, allowing a small number of initiator caspases to activate a large number of effector caspases. The accidental activation of caspases could result in apoptosis, but this is prevented by the presence of a family of proteins called IAP, for inhibitor of apoptosis. Members of the IAP family directly interact with caspases and suppress apoptosis by either inhibiting caspase activity or by targeting caspases for degradation. In mammalian cells, the permeabilization of mitochondria by BAX and BAC results not only in the release of cytochrome C, but also of IAP inhibitors. These inhibitors block the action of IAPs, allowing the cascade, beginning with cytochrome C, to activate caspases and culminate in apoptosis. The effector caspases are sometimes called executioner caspases because they are responsible for digesting more than 100 different target proteins, thereby mediating the events of apoptosis. One key target of the caspases is an inhibitor of a DNase, which when activated is responsible for fragmentation of nuclear DNA. In addition, caspases cleave nuclear lamins, leading to fragmentation of the nucleus. They cleave cytoskeletal proteins, leading to disruption of the cytoskeleton, membrane blebbing, and cell fragmentation. They also cleave Golgi matrix proteins, leading to fragmentation of the Golgi apparatus. Finally, caspases cleave and activate a scramblase enzyme that translocates phosphatidylserine from the inner leaflet of the plasma membrane to the outer leaflet. When on the cell surface, phosphatidylserine is recognized by phagocytic cells, which engulf the apoptotic cell and cell fragments.